Okay, it is my honor to introduce today's speaker and the second in our autumn series, Michael Hamblin, who will be presenting his talk on photobiomodulation. Professor Hamblin was awarded his PhD in synthetic organic chemistry at Nottingham-Trent University here in the UK, before setting up and moving his own lab to the Wellman Center for Photomedicine at Massachusetts General Hospital. He has also held the post of Associate Professor of Dermatology at Harvard Med School and is now a distinguished visiting professor at the Laser Research Center at the University of Johannesburg. Professor Hamblin's research largely focuses on different uses of different kinds of light to treat different kinds of disease, with key areas including photodynamic therapy for the treatment of cancer and infection, photomodulation, which includes low-level light therapy for wound healing, arthritis treatment, neurodegenerative diseases, as well as drug delivery, nanomedicine, and tissue engineering. This work has culminated in over 640 peer-reviewed articles with a whopping over 60,000 citations, 10 patents, and he has authored and edited 26 textbooks and holds a position at editor-in-chief at Photobiomodulation, Photomedicine, and Laser Surgery. And this work has been duly awarded and recognized. He was the first recipient of the Andre Mester Lifetime Achievement Award, Andre Mester being the pioneer of low-level laser therapy, the Outstanding Career Award from the Dose Response Society, and again, the first Ali Jaban Award for Photobiomodulation Basic Science Research. So without further delay, I'm going to hand over to Professor Hamblin for what promises to be a fascinating talk.
Thank you, Reece. Professor Hamblin, we'll be happy to receive your talk.
Get the sharing on again. All right, so I think we're all set up now. So thank you very much for the nice introduction. As was said, I ran my own lab at Wellman Center for 25 years, and I retired a couple of years ago. So I'm pretty much based in Scotland, but I am affiliated with the University of Johannesburg. So I'm going to talk about photobiomodulation today, and I have a little outline of the talk. I'm going to do a little bit about the history, because it's quite interesting how long this whole therapeutic approach has been going. Talk a bit about the mechanisms, because people are always interested. How does it really work? Talk some of our research on animal models of brain disorders, and then quite a large part of the talk will be on clinical studies and applications. So to begin at the beginning, which is Niels Finsen, who was awarded the Nobel Prize for Physiology and Medicine in 1903, so nearly 120 years ago. And if you look at his Nobel lecture here, you'll see that he had Niemann-Pick disease. And when he was young, he found if he went out into the sunshine, he got a lot better. And that motivated Niels Finsen to think that light could treat all sorts of diseases. And here is one of the books he published. And he basically developed lamps, electric lamps, arc lamps, incandescent lamps to treat all sorts of conditions. And after Niels Finsen, this motivated a lot of folks to think about light therapy, because getting a Nobel Prize is a pretty big deal. And there were several books written. And interestingly enough, because electric lights were just really becoming universal, and because Edison has invented the light bulb, and John Harvey Kellogg of Kellogg's Cornflakes fame, he believed in a lot of natural therapies, and he devised these electric light bars with bulbs in, incandescent bulbs, different kinds of bulbs, but treated all sorts of diseases and conditions with these electric light bars. Pretty much at the same time, people began to think about sunshine as a therapy, and this was called heliotherapy. And the clinics that started to be open for heliotherapy were up in the mountains, quite often in the Swiss Alps. And here you see lots of folks outside in the snow, exposing themselves to sunshine. And Auguste Rollier, who had a clinic, so that if you just lie on the beach, all you get is a hot air bath. But if you go up in the mountains, you get a medical therapy. And he thought it was the temperature, but in reality, I believe it was the low oxygen tension that you get up in the mountains that made your mitochondria more susceptible to the effects of the light. And then fast forward a few years, and the race for the first working laser in the 1950s, people may have read about that, but this race was won by Ted Neiman in 1960, who built the first working ruby laser. And pretty soon after that, Paul McGuff, who was working in Boston, treated tumors with the ruby laser. And here he had a paper in the New England Journal of Medicine showing that a ruby laser beam could cure not only experimental tumors, but actually human tumors. Here you see a picture of the ruby laser beam in an operating room. And then very soon after that, Andre Mester, who was working in Hungary, developed an interest in laser research, and he got a ruby laser, tried to repeat Paul McGuff's experiment. But because his laser was only a fraction of the power of McGuff's laser, this didn't work. And then he said, right, okay, let's see if we can cause skin cancer with this laser beam. And that didn't work either, because light doesn't cause skin cancer, at least not this kind of light. But he did observe increased hair growth and increased wound healing in his mice and his rats in the laboratory. And this started off a huge thing that lasted even today, which was really called low-level laser therapy. And maybe people haven't heard of photobiomodulation. I'll just tell you why we changed the name. So it turned out you didn't need a laser. Originally, people thought there was something magic about laser beams. It could be the monochromaticity, it could be the coherence, but there was something special about lasers. But since the vast proliferation of LEDs, we realized there's nothing special about a laser, so get laser out the name. And then it was low level. Well, nobody really knows what low level means. It's a completely relative term. And because some of the applications do not involve stimulation, they involve inhibition, like inhibiting pain signals, for instance. So that is why the whole field decided to call it photobiomodulation. Okay, so getting on to the mechanisms. For a long time, it was realized that mitochondria were one of the big players in the cellular mechanisms. Mitochondria, of course, the powerhouse of the cell. People did experiments and isolated mitochondria and lots of mitochondria inside cells with various kinds of fluorescent dyes and other ways of assessing what changed in the mitochondria. So mitochondrial oxygen consumption went up, the mitochondrial membrane potential went up, the ATP produced went up. Well, you got a lot of signaling. So cyclic AMP went up, nitric oxide was released, a brief burst of reactive oxygen species, and changes in calcium. And it began to realize that the mitochondrial metabolism changed from glycolysis towards oxidative phosphorylation. All these changes in the mitochondria, the ATP, cyclic AMP, the nitric oxide, the ROS, they can activate things like Jun-FOS and NF-kappa B, which are very pleiotropic transcription factors. So these transcription factors can affect the expression of well over 100 genes. So there's a lot of changes in the proteins that are produced in the cells, growth factors, extracellular matrix proteins, proliferation, motility, anti-apoptosis, and pro-survival. So a single exposure in the laboratory to light, like in mice, we have a mouse model, a single exposure of mice will have major effects that last up to 4 weeks. So, you know, while some of the experiments you get brief effects when you expose the cells to light, in animal models, it's possible to have very long-lasting effects. As I said, you get this switch from glycolysis to oxidative phosphorylation. This has 2 major implications, which are not immediately obvious, but stem cells in the hypoxic niche carry out glycolysis because there's no oxygen in the hypoxic niche, so they don't have a choice. But when the mitochondria are activated, the stem cells have to leave their niche in search of oxygen. And once they get mobilized, they encounter cues depending on what is wrong with the tissue in the body, if it's an injury or degeneration, and they activate their proliferation and differentiation programs. So to some degree, photobiomodulation can be considered a type of autologous stem cell therapy. Macrophages have an M1 phenotype. These are the pro-inflammatory macrophages that, you know, get chronic inflammation. Then they carry out glycolysis. But when oxidative phosphorylation is activated, they switch their phenotype from M1 to M2, which is an anti-inflammatory phenotype. And especially in the brain, where the macrophages, of course, are microglia, the M2 phenotype can carry out phagocytosis, for instance, removing amyloid plaque in the brain, but doing lots of other beneficial healing type functions. So that explains why photobiomodulation is anti-inflammatory. In addition to mitochondrial metabolism, there's another type of photoreceptor that's quite important. It's just beginning to be understood. And it's these TRP ion channels. So funnily enough, the Nobel Prize awarded 2 days ago was for the discovery of TRP ion channels, which are capsaicin and heat-sensitive ion channels. But some of these ion channels are also light-sensitive because they have opsins, which are light-sensitive proteins. And others are heat-sensitive, so you get a conformational change. And the effect of activating these ion channels is you get big changes in calcium, which also happens in the mitochondria. So the fact that the mitochondria are involved can be explained by mitochondrial chromophores and by heat-light activated ion channels. As a third mechanism that's been proposed by some people, so in the mitochondria, ATP synthase, which is unit 5 of the mitochondrial membrane respiratory chain, it produces ATP. It's actually a molecular rotor. And the idea is that it has nanostructured water, and when this water absorbs light, the viscosity is reduced, and the little rotor can whiz round faster and make more ATP. One very hypothetical mechanism that's just started to be proposed is that some wavelengths in the near infrared, for instance, 1270 and 1064, can directly excite ground state oxygen, which is triplet, to form the singlet oxygen. Here you see the absorption spectrum of oxygen. And you probably need fairly intense photon density to excite oxygen, but it does explain why 1064, which has been used in the brain, may work. But again, that is a little bit hypothetical at present. It was recently discovered that blood contains circulating cell-free respiratory competent mitochondria. And here you see, does the blood we thought to know so well contain elements that had been undetectable until now? Because you know, people have studied blood for hundreds of years. Just in 2019, they found the cell-free mitochondria, but it does explain why photobiomodulation can have a significant systemic effect. So if you shine light on one part of the body, it can have effects on distant parts of the body that didn't receive any light. So there's something circulating around that can be activated by light and be beneficial. Nevertheless, most people think that the light should go into the tissue where you want to treat. And that obviously depends on the wavelength. So here you see the tissue optical window, absorption and scattering. There's a little window in the red and the near infrared where the light can get in deeply. So if you want to treat, you know, deep lying organs or joints, you need to choose the right wavelength. It's also a biphasic dose response. So, you know, you give up some light and you think, good, this is working, give a bit more. But if you keep giving a bit more light, the benefit actually goes away. And even if you give a huge amount of light, you might do some harm. So you need to think a little bit about getting the dose right. Okay, so that was the history and the mechanisms. In the animal studies, we had funding from the US Department of Defense to work on traumatic brain injury, which was a big interest of the DoD. And the first study we did, we had a model in mice of closed head TBI. So basically we dropped a weight on the head of the mice. And in the lab, we had 4 different lasers. So we thought, right, you know, we're traditional photobiologists. We want to see some evidence of an action spectrum that different wavelengths do different things. And this is what we found, that red laser, 665 nanometer, a single exposure to the head of the mice 4 hours after we hit them on the head, just one treatment. And we assessed their neurological performance for 4 weeks. You know, so it's a huge difference. And another laser, 810 nanometer laser, exactly the same results. A single exposure to the head 4 hours after we dropped the weight, and they steadily improved. But 2 other wavelengths, 730 nanometers and 980 nanometers, we saw no positive effects at all. So that reassured us a bit, because we like to see, as I say, some kind of action spectrum doing these studies. However, dropping a weight on the head of mice is a little non-reproducible. So then we got a special machine to cause a TBI in mice, and you actually have to open the skull, and then there's a controlled piston can impact the brain. And again, we used the 810 nanometer laser, but this time we used different pulses. So we used the same dose, which was 36 joules per square centimeter at 50 milliwatts per square centimeter. One dose, 4 hours after the injury. We compared pulsed at 10 hertz, pulsed at 100 hertz in CW. So all these different pulses worked pretty well, just as we saw before. But interestingly, the 10 hertz worked significantly better than the 100 hertz in the CW. And people now think that the reason for this is due to the alpha rhythm of the brain, which is the 10 hertz electroencephalograph rhythm, and pulsing the light can resonate with the natural rhythm. So that is the theory to explain that observation. We wanted to see if this approach could do something better than just improve the neurological performance, which was just like balancing and finding a way out of a circle. It wasn't really any intellectual brain power. Not that mice are great intellectual brain power anyway, but never mind. You can measure learning and memory in mice, and you do that with the famous Morris-Water maze, where the mice have to remember the visual cues to find a hidden platform. And here you see that with what we did here was compared one laser treatment with one a day for 3 days, because we thought, well, nobody's really just going to get a single laser treatment. We had to turn the dose down because one worked so well that in order to see a difference, when we cut the dose down, and 3 then worked better than 1, but they both significantly improved the learning and memory compared to the untreated TBI mice and to the. Then sacrificed the mice, took out the brains and tried to find out what was going on in the brains. And we wanted to look for neuroprogenitor cells. So these are new brain cells, and some people call them neural stem cells, but these are newly formed brain cells, and they are BRDU UN double positive. And 7 days, you see these little blue cells here. These are the double positive cells in the dentate gyrus of the hippocampus, which is where your new brain cells are made in the dentate gyrus. It's 7 days. We then looked for some other markers in the brain, and possibly the single most important molecule you can have in your brain is BDNF, brain-derived neurotrophic factor. And here you see this is the dentate gyrus and the subventricular zone. Again, at 7 days, you get these red positive BDNF positive cells. Here's the graphs showing that it's 7 days, but not 28 days. It's gone by 28 days. Now this is synapsin 1 staining. So synapsin 1 is a marker of new synaptic connections. So there's a difference between newly formed brain cells, which is neurogenesis, and newly formed synaptic connections, which is synaptogenesis. So here we find significant increase in synapsin 1 staining, this time at 28 days, not at 7 days. And the biggest was in the perilesional cortex. So that is the actual main part of the brain, not particularly the dentate gyrus. This is where the action is in the cortex. So it looks like something happens at 7 days in the dentate gyrus, and then that stimulates something happening in the cortex at 28 days. Then we looked at a specific mouse model that had a defect in this IEX1 gene. And these mice, when you gave them a brain injury, had much worse outcomes. So the mice, these were genetically engineered mice, but they had much worse outcomes when you hit them on the head, basically. And again, the photobiomodulation, again, we called it LLLT in those days, for the knockout mice had a much bigger effect than it had on the wild type mice. So the fact that these mice have this genetic aberration means that their performance is worse when you give them a head injury, but their improvement is much better, relatively much better with the light. It's very visible here. So knockout mice, this is like a gap in the brain, and it's pretty much completely prevented when you give them the light. I also looked at, could you combine this with metabolic modulators? And this was a in vitro study showing that when you add these modulators into the cells, you get a much bigger effect of the light than So this is the in vivo study, again, with these metabolic modulators. And again, you get a bigger effect of the light in the TBI model when you combine it with just simple metabolites that are used in the mitochondria. Pyruvate is the main one, but lactate sort of works a little bit, but pyruvate is the main one. So this is a study looking at sleep deprivation. So here is a tank of water and little platforms. Now, if the platforms are big, the mice are quite happy. They sit on the platform and they can go to sleep and they're quite happy. But if the platforms are tiny, if the mice go to sleep, they fall off and they don't want to fall off, so they don't go to sleep. So these are sleep deprived mice, and that gives them cognitive impairment and oxidative stress and hippocampus. So this shows that when you give the light to these sleep deprived mice, there is a significant improvement in various cognitive measures. This is escape latency from the maze, a what, where, which test, the Barnes-Maze test. So that the light goes back to baseline, basically. So these are the sleep deprived. And you get big oxidative stress, but the light reduces the oxidative stress in the hippocampus. This was a neonatal rat model of hypoxia ischemia. And you probably know that a lot of babies get brain damaged at birth because something happens during the actual delivery and oxygen supply to the brain is interrupted. And these poor kiddies get, all sorts of lifelong cognitive impairments and cerebral palsy and all sorts of horrible things. So, if a baby was to get into difficulties into birth, could you put near-infrared light on its head right away? Something worth thinking about. So this was an animal model in rats. And again, the photobiomodulation had significant effects in various cognitive tests, object recognition and Barnes-Maze. So the newborn rats that had their oxygen supply interrupted immediately after birth could be partially rescued by giving them near-infrared light on their heads. We did a systematic review of Parkinson's in animal models. It didn't actually work in Parkinson's models, but several other people have. And the answer is that photobiomodulation works just fine in animal models of Parkinson's disease. So there are many, many mechanisms of photobiomodulation in the brain. I've kind of arrayed them all here so that we showed the neurogenesis, we showed the synaptogenesis, we showed the neurotrophins. Other people have showed anti-inflammatory, antioxidants, increase in superoxide dismutase, anti-apoptotic, more survivin, more BCL2, cell migration, angiogenesis, less edema, because when you have a head injury especially, you get edema. That's terrible, because you increase lymphatic drainage, which can reduce the edema, and increased blood flow and cerebral oxygenation. So a lot of beneficial things go on in the brain when you shine near-infrared light. And because the animal models are so convincing, a lot of people have done clinical studies way before we started working on it, actually. But so this is photobiomodulation overall, and you can see that it's pretty much any condition that you can have. I mean, I wouldn't say it's great for infections, but it is good for COVID because COVID has, you know, cytokine storm and systemic inflammation, and photobiomodulation is quite good at reducing systemic inflammation. But people have used it for lung inflammation in the past. As I say, I'm going to talk about the brain applications in a bit, but a lot of dentists use it for, you know, TMJ, dentistry pain, oral mucositis, allergic rhinitis in the nose, helps the ears, deafness, also helps the eyes. People say, oh, can I shine this light in my eyes? And the answer is it's actually good for your eyes. It will preserve your eyesight. And for dry AMD, it's been approved as a treatment, which is one of the few treatments. So a lot of pain things, neck pain, tennis elbow, carpal tunnel, fractures. If you break a bone, it doesn't heal. Heart attack is interesting because you can't easily get the light to the heart, but it does work, possibly via mobilizing stem cells, as I talked earlier. Kidney failure, kidneys are not too deep, so we get to them fairly easily. Lower back pain, clearly that's a big deal. Arthritis, a big deal. Muscles, the muscles have huge numbers of mitochondria, so it's a big deal for muscles. Tendinitis, one case, wound healing, diabetic ulcers, all sorts of things like that. So it's difficult to find, you know, especially injuries and degenerative conditions that can't be helped by photobiomodulation. So these are the laser systems that were used because it started off as laser therapy. You know, for many, many years, there were laser therapists that had their sort of portfolio of lasers, if you will, their armamentarium of lasers, and they thought they were able to judge keenly which wavelength, how much power, what pulse duration. It was a bit of an art form, but nevertheless, these laser therapies still going. They do a lot of good. When LEDs came in, this idea of whole body photobiomodulation kind of got popular. So Novo Thor's a whole body light bed. There's several of these available now. Big panels you can stand in front of to get a lot of light into the body. Novo Thor is 500 watts of optical power. And, you know, if you could get a 500-watt laser, that would cost you millions of dollars. Anyway, there's home uses because LEDs are harmless, so you can get a lot of LED devices to use at home. Some of them are flexible, so you can wrap them around joints and various things. Some of them are handheld, put them on a stand. I certainly think that, you know, in the near future, every household will have at least one LED device for, you know, painful conditions and injuries and wounds and these sort of things. And a lot of devices being studied to put on the head. And some of them are a bit smarter than these. Some of these look a little bit sort of Heath Robinson, you know. So there are companies now making sophisticated helmets that you can put on the head. But because the effect on the brain is so pronounced, this is going to be an even more rapidly growing area, in my opinion. So one of the first clinical studies we did was with Neilinson in Boston, and she used this near-infrared red LED cluster to put on the forehead. And she had two ladies who had chronic TBI. One was 7 years after a traffic car crash, and after 8 weeks, her ability to do computer work had improved tenfold, and she got a home unit and has used it continuously for many years. Another lady had multiple concussions and PTSD, and Marnie gave her a home unit, treated every day, and she improved with some objective tests of executive function. Stroop test, this was the significant improvement in the Stroop test. That's the, where they give you words of colors and the color of the font doesn't match the name of the word. So you see the word red, but it'd be in green font, and you'd have to not get confused. And these were memory tests for recall and delayed recall with significant improvements. So Marnie was excited in this and got a series of 11 patients with chronic TBI. And out of these, 1, 2, 3 didn't really respond, but the remaining 8 patients had significant life-changing improvements. And some of them who had been off work for a long time could return to work. And this one couldn't speak, but became quite chatty after the LED. So makes a difference to these folks. This was a randomized controlled trial that was run at Mass General Hospital, and it was a difficult trial to run because patients who are brought into the emergency room at Mass General are leapt upon by all sorts of people to recruit them into trials. So there's a lot of competition for patients. And the other thing we didn't realize is that a lot of them are sent home relatively quickly. So originally we said, oh, we're going to treat them once a day for 5 days, but a lot of them were sent home on day 2 or 3. So that was another difficulty. But nevertheless, we did see an improvement in various TBI scales. There's a clinical symptom scale that they use, and the light treatment had a significant improvement. So this was the BioLight Neuro, BioLight's a small company in Toronto in Canada, and they have this device which has 4 LED clusters designed to illuminate different parts of the brain. So this is one of the big questions that's still not completely settled in brain photobiomodulation. Do you want to put the light all over the head or on the forehead, because there's no hair? So light goes into the forehead better than if you put it on top of the head where there's a lot of hair. Or do you want to try and target specific parts of the brain? So this was moderately severe dementia. I mean, not all of them were diagnosed with Alzheimer's. Probably they all had Alzheimer's, but you need specific diagnostic criteria for Alzheimer's. But nevertheless, they were treated twice a week for 12 weeks, and there was an amazing increase. This was about 7 times higher than the increase in the scale that they got with the large clinical trial for Aricept, which is the only really approved drug for Alzheimer's. It doesn't work very well, but photobiomodulation seems to work a lot better. So after 12 weeks, the treatment was stopped and their cognitive performance deteriorated. But after 16 weeks, they then were given a home use device, because it's a simple little LED cluster, and they, not on this chart, but they actually improved for a second time when they were given the home use device. So it's likely that when it's used for treating Alzheimer's, it's going to have to be used for the rest of your life, basically. This was another single case report, but it was quite interesting. They use 3 different kinds of light: this sort of hood, an intranasal device, and also a pad they put on the belly. It's not shown here, but I can't see it because it's in here. But these people had a big improvement in various things that were measured. Attention, executive function, but also the smell test increased dramatically. And you probably know that if you have Alzheimer's especially, or any sort of dementia, your sense of smell tends to go. But this can bring it back. And also, in some of these things, you give a questionnaire to the caregivers and they improve. In fact, in the Violite trial, the caregivers started to use the device themselves because it's a lot of burden taking care of a dementia person, and it helped the actual caregivers. So this was depression and anxiety. Quite a long time ago now, but Fred Schiffer, again in Boston, had an LED device on the forehead, and he also measured the near-infrared spectroscopy for brain oxygenation. And this shows that the light dramatically increased the brain oxygenation inside the head. And two scales, Hamilton scales, this is depression, this is anxiety. So there's a single treatment, and 2 weeks later, all the patients dramatically improved on both scales. 4 weeks later, it had worn off, but they only got the one treatment actually. And again, you know, it's like TBI, like anything, you probably are going to need to give more than one treatment. So Paolo Casano, again in Boston, he's a psychiatrist. He started to look at it. This was a laser on the forehead, and he gave 6 treatments, and the gradually improved, steadily improved over the course of 6 weeks, now with significant improvements in depression scores. And Paolo now has a clinic at Mass General treating depression, you know, who are resistant to antidepressant drugs. And there's a certain percentage of patients that are resistant to antidepressant drugs. And he has a clinic to treat them with photobiomodulation. We haven't worked personally on Parkinson's disease, but there are a group working in Australia who have these so-called light helmets or light buckets, which are simply lined with lots of red near-infrared LEDs, and they put them on the head, 20 to 30 minutes twice daily. And you know, for Parkinson's, there's all sorts of symptoms, but these are 4 patients. And if you just look overall at the colors, if they deteriorated, it would be in red, and there are no red colors at all. Yellow means no change, so a few of those, but green means they got better in this particular symptom, and the vast majority of these are colored green. So again, it seems to have major beneficial effects on Parkinson's disease. And the Australian folks are running trials at the moment. I don't think they've actually finished them. But this caught quite a lot of press attention. Is infrared light the answer to Parkinson's? And they also have this idea that it can help the gut microbiome. So in addition to putting the light on the head, they put it on the belly. And the idea is that the photobiomodulation can improve brain function by putting it on the head, and can improve the gut microbiome by putting it on the belly. But again, that really needs more work to confirm that. Again, Marnie Naser and Linda Chow, they had got a grant to look at Gulf War illness, which is a big deal in America. So a quarter of the US veterans in the Gulf War came down with this Gulf War illness. So they use this Violite Neuralfats as the little headset I showed you before, 3 times a week for 12 weeks, and got clinically, that's a previous slide. Anyway, they got clinically meaningful benefits in the Gulf War illness. This was another study from Fred Schiffer, and he looked at opioid craving. So basically drug addicts, he used the same one he used before, the little LED cluster on the forehead, and the active 53% improved, the sham was only 20% improved, and this was significantly different. This is Agnes Chan, who in Hong Kong. So she's working on not necessarily people that are diagnosed with a brain disorder. So this was just elderly folks, as you get older, your brain function is not all it used to be, especially with things, memory and reactions probably. So again, she put the light on the forehead, various points on the head. And so this is a category fluence test result. So what this test is, you are given a certain time, like 30 seconds, and you have to name as many four-legged animals beginning with B as you can, something like that. And the ones that had the active photobiomodulation could name at least 3 times as many categories in this time as the ones that didn't. So that made a big difference. Another thing that Agnes did was gave these people a difficult cognitive test. Again, real or sham, 810 nanometer LEDs to the forehead, single exposure. And she also was able to measure the oxygenated blood flow by this device. So it had the LEDs for the treatment and also the LEDs that can measure the oxygenated blood flow. And what she found was they didn't get any better at the performance, but what did happen is they used significantly less oxygenated blood to get the same performance. This is the control and this is the experimental. So made a difficult cognitive test less arduous, as she said. These were folks that had mild cognitive impairment, 3 case reports, visual memory, verbal memory, some major improvements, especially here. Big improvements in cognitive function. So a lot of common pathways in neurodegenerative and psychiatric disease. As I say, BDNF is hugely important. Neurogenesis is important. Synaptogenesis is important. If you let these things progress, does your hippocampus in, and if you let that progress, your entire cortex shrinks. And neuroinflammation, oxidative stress, excitotoxicity, mitochondrial dysfunction, neuronal apoptosis, all of these are linked in with these bad things that can happen in your brain. And they are caused by Alzheimer's, Parkinson's, depression, PTSD, bipolar disorder. People haven't really tried bipolar disorder yet, but I'm sure they will. And also schizophrenia, which are major psychiatric disorders that could possibly be benefited by photobiomodulation. Listed some of these brain disorders here, a lot of traumatic things, stroke, obviously, TBI. If you have a heart attack and the blood supply to your brain is interrupted, you only have a few minutes to get it started again, or else you'll get brain damage. I mentioned birth trauma, and can putting near-infrared light on the head bring people out of a coma? There's some indication it might do. Autism and ADHD, I haven't shown any data, but people are working on these, and especially with autism, they're getting remarkable results. Again, PTSD with some animal studies, also some human studies there. Addiction, I mentioned that. Insomnia, it's great if you have problems sleeping. Put some near-infrared light on your head before you go to bed, you sleep like a baby. There's a lot of neurodegenerative conditions aside from Alzheimer's and Parkinson's. Marnie's working on CTE, which is what football players and boxers get. There's a little bit of work done on Huntington's disease. So people sometimes think, is there any overlap between photobiomodulation and brain stimulation? Because they're used for some similar brain conditions. So non-invasive brain stimulation implanted electrodes of the classical way for Parkinson's, deep brain stimulation, but repetitive transcranial magnetic, transcranial direct current, and even low intensity focused ultrasound applied to the scalp. All these can treat these brain disorders. So there is some overlap. I think it's not exactly the same because a lot of these non-invasive brain stimulations will trigger action potentials in the neurons, and that doesn't happen with light. So, you know, if you get a TMS on your head, immediately your arm will jerk, but that doesn't happen with light. So it's not quite the same. We published a book about photobiomodulation in the brain. Again, it's a rapidly growing topic, so a little plug for that. So the conclusions: photobiomodulation has a history of over 100 years. The mechanisms are becoming understood. We've done a lot of animal studies of TBI and other brain disorders, and we collaborated with clinicians, acute and chronic TBI, Alzheimer's, depression, opioid addiction, and cognitive enhancement. So that's pretty much it. This was my lab just before I left Boston. They all moved on to other things. Various funding agencies who funded this, and thank you all for listening.
Michael, thank